right. Good morning, everyone. Actually, good afternoon, I guess. I always want to say good morning. I'm so used to morning time, but it is definitely not morning anymore. So before we get into class topics today, I wanted to take just a moment and talk briefly about our written assignment. I think I mentioned to you guys that I had posted it on our Moodle, but we didn't go over it yesterday or Monday because I wasn't sure how much time we had left. I have done the math and we can now afford to talk about it just a little bit. So that's what we're going to start off with this morning. So this is the description that's up on Moodle right now. So you might have already read through that, in which case you can zone me out for a couple of minutes here. But the point for this written assignment is to get you to think critically about how a popular media article talks about a scientific research study. So what I'm going to get you to do is look for a popular media article, either a newspaper, a magazine, a website, BuzzFeed, whatever. Something that talks about a research study. A really great place to start would be searching for something like scientists prove that, or scientists show that, and look for things of ill repute, sites that we wouldn't necessarily trust right off the bat. And so in this media article, you're going to be looking for something that's going to give you an indicator of what study they're talking about, whether they tell you the author's names, the title of the study. Maybe they're really good and they actually link you to that scientific article to look yourself. That's the ideal case. And so what this assignment is comparing that popular media article to the original scientific research. And don't worry too much. You don't have to read the whole scientific study. You can get most of the information that you need just from skimming the abstract, which I stress here because I have had students in the past worried about trying to read very dense articles. So that's not the focus here. So you're going to be choosing an article and associated research paper that let you answer the series of questions that I have here. So the first one is, what is it that the popular media article is claiming? What do they say that the research has found? What are they talking about? So this is when you give me a general description of what that media article is saying. Then the second question is, how confident are you in those claims? So thinking critically, talk a little bit about, does this seem believable? Are they following some of those scientific concepts that we talked about in chapter 2 and as we've been talking about different kinds of research? Or are they doing something bad, like talking about causal relationships after doing a correlational study? So this is that find good or bad, what makes you think that they're telling the truth or not? And then the last point is to go to that abstract of the original scientific research paper and see what the research actually found. Are there any differences? Did the popular media article do a good job talking about it, or did they maybe stretch some claims? Did they leave out information? That sort of thing. And so you'll notice here that each of these questions is listed with a point value. So question 1 is 1 point, 2 is 3 points, and 3 is 2 points, which work out interestingly. But the way that I do it is when I'm asking you for a point, I mean like write a little literal single point, make a single argument, make a statement. So for a question that requires 1 point, you could write a single sentence, and as long as your sentence makes a good argument, you get the mark. For the one that's worth 3 points, maybe write 3 sentences. And if you include things like compound sentences, that's fine too. I'm not going to be counting periods, I'm counting ideas that you're presenting. So if you're doing the math here, you can finish this assignment in as little as 6 sentences and get full marks. This is why I tell you not to stress too much about the assignment before that first midterm, because we still now have over a month to work through this. So you're going to spend a little bit of time finding an article that points to research that you can use for this. But once you've done that, it's not intended to take a really long time. I'm looking for you to show some critical thinking, to put some ideas together, and to evaluate how good of a job the media article has done. So maybe it's done a great job and they've simplified the science and presented it in a more accessible way to the public. Or maybe they went with a clickbait title and they're making invalid claims. That's what you're going to tell me about those two articles. And then the last thing that I'm asking for is an APA citation. So I want one for both the popular media article and the original scientific paper. And underneath each of those, I also ask you to give me a hyperlink that I can click on that will take me to both of your articles. And that's just so that I can do a quick check that your citations are correct. And maybe if I have any extra questions, I can look at the articles themselves. So if you haven't done an APA citation before, the easiest way to do this, and the way that I do it every single time I need to do one, is I go into Google and I type in APA citation. And there are two things that always come up right off the bat. The first is the APA Style Guide. So that's the American Psychological Association's official website. And I hate it, so I never use it. Instead, I go to the second option, which is the OWL Purdue site. Same information presented in a much more logical format. That's what I use. Um, and if you've done this before, you might have it all memorized and don't need a reference. When I started, we were, gosh, actually, we might have been two editions back. We're now on the seventh edition. I did my thesis in the sixth edition, but I'm pretty sure as an undergrad we were doing fifth edition. So there have been some changes since, which is why I always pull it up. So when you're doing it, I recommend doing that. And they show you examples of how would you arrange the author's names, the title, the article number, that sort of stuff. What order do you put them in? Is it italicized? Is it not? All of that stuff, they walk you through it. And I'm going to be looking for a good video to link that walks you through an example of this. If I can't find one that I love, I'll just put myself. But I do find there are experts on this who can explain it a little bit more clearly than I can. But yeah, so that's the only part that needs to be an APA are those two citations. The rest of it are going to be answering those three questions. So this should be able to fit onto a single page. If you feel like you need a couple of extra sentences to get your point across, you can. I don't have sort of a maximum or minimum word limit. I'm just looking for you to make the point that you're meaning to. Yeah, so aiming for less than a page. I'm not, don't have any hard numbers. I would like your questions to be numbered so I can see where they are and what they're talking about. That really helps. And when you're done, just save it as a PDF and you can upload it to Moodle. I already have that written assignment submission portal there, so you have access to that already. I recommend you submit early. Don't leave it to the very last minute. I do tell people that even though there is a hard cutoff, if you're a few minutes late, I'm not going to dock you a full day's worth of penalties. So if something goes wrong with your computer and it's 30 seconds late, I'm gonna just ignore that. So usually up to 2 or 3 minutes, I don't mind. And then there are deductions for longer lateness. So the way that I've worded it is it's a 10% deduction for every day that you're late. So if you are an hour after the deadline, that's the first day. So that's a 10% deduction. If you are 26 hours after the deadline, that's 2 days. So that's 20%. With the caveat that if you can contact me in advance and let me know if something is going on, I don't mind moving a deadline for individuals who have a valid reason. So I think that's all of the important information there. If you have any questions about that, or if you think of anything, let me know. Anyone have questions that have popped into mind already? Yeah. This is just the written assignment. So what, I don't remember what percentage it is because all of my classes have one and they're all different weights. So let's do a quick check. So it is 10%. Yeah. And if you have, like, as you're working on it, maybe if you want to check your answers, if you can get in touch with me in advance, I don't mind looking over it and dropping some hints. Awesome. Okay, so that was the written assignment. I don't think I had anything else I wanted to say about that. So we can now hop right over to the last of our topics for this chapter, and this is our visual system. So I mentioned earlier, I do this last, even though most textbooks do it first, because now we've kind of built up the right vocabulary to talk about things. We understand some of the concepts that we need to talk about this. And right off the bat, you're going to start seeing some of the parallels to our auditory system as we start talking about how the visual system works. So as per usual, we can start with what is our stimulus? And here we're looking at light as our stimulus, and it's a form of electromagnetic radiation that travels as a wave. And what that means is that light is a particular wavelength of this radiation that we are able to perceive with our eyes. That same electromagnetic radiation, if it has different wavelengths, if they are smaller, we can have things like UV or X-rays. If they are larger, we can have things like microwaves and radio waves. So those are all the same kind of energy, but with different wavelengths. And if we look at the scales here, we're looking at sort of fractions of a millimeter all the way up to many, many kilometers. So they vary quite a bit here. And if you've studied light, especially if you've done something with physics, you might know that light can be both a particle and a wave. We kind of ignore the particle aspect and care about its wave properties, and then hand wave the fact that it interacts with receptors. So another one of those topics where we could go into a whole lot more detail about the atomic level of what's going on, but we're going to choose not to. And so for our visible spectrum, what we can see is going to be somewhere in the range of 400 to 700 nanometers wavelength that we're looking at. And so different species will have different abilities to perceive things based on what kind of receptors that they have. For example, birds, most species of bird can actually see into the UV spectrum. So on that blue end of our spectrum, they have even smaller wavelengths that they can see, so then they can see UV, which is kind of neat. All right. And so when we're talking about those properties of a wave, this is where we see that overlap very extensively with our auditory system. So we can look at amplitude and wavelength and purity, which we had already looked at when talking about sounds. And so here, amplitude, that sort of deviation from the center of our wave, higher amplitude in sound was more energy and a louder sound. In light, it's more energy that we see as more brightness. So brighter light because there's more energy there in the wave. We can also look at those wavelengths. So longer and shorter wavelengths have differing colors. We perceive them as different colors. Ooh, wrong way. So the smaller wavelengths, the 400 nanometer range, that's that blue-violet end of the spectrum. And our 700 nanometer range is the red end of the spectrum. So the different wavelengths correspond to different colors that we'll interpret the light as being. And then our last one is purity, and that's whether our waves are sort of pure and sort of a single, very clean wave, or are they messy? Are there multiple things mixed together? In sound, purity was that quality of the tone. So a pure sine wave would give us a very clear sort of buzzing tone at a particular frequency. So with purity in light, pure light would give us a very highly saturated bright color, and a very impure or messy signal would give us an unsaturated color. Lots of noise, they look a little bit more gray. So those are our similarities between light and sound in terms of describing those waves. So the next thing we're going to do is talk about the actual process of seeing and all the structures involved in that. So this is going to be sort of on that detail end that we saw with the sense of hearing when we were looking at the structures of the ear. We're not going to go into all of the details, but we're going to hit the major points required for understanding how this goes together. So I'm going to start by doing a single walkthrough on sort of one big image, and we'll talk about light moving from sort of outside the eye into the eye and then passing to the brain. And then we're going to have slides that go through it one more time, but with words. So we're going to do it twice, just in case it doesn't sink in the first time. And I always find that some people do better with pictures, some better with words. So we're going to do a little bit of both. So starting with our image here, we're going to start from the outside. So on your left, the first thing that the light is going to pass through is the cornea, and that is that outer layer on our eye. So that's clear, that allows light to pass through it. And then in order to continue onward, it's going to move through our anterior chamber that has our aqueous humor. So that's the first of our sort of squishy liquids that light passes through. And then we pass through a gap, and it's not labeled here, but that gap is our pupil. So that's the dark part of our eye. And the pupil is dark not because there's a dark substance there, but because it's a hole, there's less light inside our eye than in the environment around us. So it looks black because of the lack of light. And the size of our pupil is determined by the iris. So the iris is a ring of muscle that will expand and contract to let more or less light into the eye. More light coming in will mean that we're getting more light that we can see. But if the environment is really, really bright, you might have noticed that your eyes have very small pupils because you don't need all of that light. It can be overwhelming. So those irises will adjust so that we have the right amount of light to see properly. All right, so through the pupil, the next step is our lens. And the lens is just what it sounds like. It is a structure that will bend light in a particular way so that it hits the back of our eye. And the lens isn't like a solid piece of glass like in our glasses, but it's something that's movable. And our suspensory ligaments are involved in holding up and supporting that lens, but they can also stretch it and adjust its size and shape. So it'll get sort of taller and skinnier or shorter and fatter, whether we're trying to look at something that's close or far away. And so once we've passed through the lens, the light's been shaped, and it's going to hit the back of our eye. And the back of our eye, we're looking at the retina. The retina is basically like the screen that the light is going to hit, and that's where our receptors are. Those receptors are going to send that image to the brain. And it's going to send that signal via the optic nerve at the bottom there. So that's where our neurons are going to come together as the optic nerve, all bundled together, and head off into the brain to send that information to our occipital lobe, where we can start processing. The one other feature that we want to point out here is the fovea. And the fovea is our focal point. So that's where we have a really high concentration of receptors. That's where we get the most detail in our vision. So that's going to be where our eyes look naturally. So almost exactly back from where the light would be entering into our eyes. So it's sort of straight back behind and a little bit above, and that's where we get our best vision. All right, so as I said, that is a lot of steps with no words. So let's do it one more time now with the words and remembering what that image looks like. So we go back to our steps here where we have light entering through our cornea. It passes through that hole, that's the pupil, and it's going to pass through the lens. The pupil isn't necessarily a structure, it's sort of a lack of structure, which is why it isn't included in sort of the steps there. I'm just reminding you where that happens. And those adjustments that we were talking about with our lens, that stretching and squishing, we can now call those by the proper term, which is accommodations. So those adjustments that are made that allow us to focus on things that are closer or further away are accommodations. So when you focus on a close object, the lens is going to squish down and get more round and fat. That's going to let us see things that are up close. And when we focus on distant objects, the lens is going to stretch out and get tall and skinny. And we can have problems with those adjustments. Some of us wear glasses. I can't see things that are up close, so my lens doesn't squish enough when I'm looking at things sort of in this range. So my glasses help correct the kind of adjustment that I can't do on my own. And when we're talking about sort of adjustments and slight changes that our eyes can make, we can also look at something called saccades. And these are tiny, almost imperceptible movements in our eyes. Maybe you've been having a conversation with someone and you notice that their eyes just shimmy a little bit back and forth. So there's a slight, very quick movement that happens in our eyes, and it happens all the time, whether you're aware of it or not. And these slight movements actually help us have better vision by focusing on sort of different parts of the same object. So it gives us an idea of how far away something is and a better idea of the shape of it. So we're moving around in the environment, kind of hopping between things. So this is critical for us having good vision. If you're also watching someone's eyes very closely, these little darting movements, they might bias towards something that they're trying to look at without telling you that they're looking at it. So they can give away intention if you're really good at reading them, though most of us are not. Wrong way. Doing that. Okay, so that's the sort of front part and the lens, and then we're looking at the back part, which is our retina. So the retina is absorbing light. The retina also does some low-level processing. So we start doing some processing even before these signals reach the brain. And what's going on here is that we have so many more receptors than we could afford to have neurons leaving the eye. So we condense the signal. So instead of each and every single one of our receptors getting their own neuron, some of them are going to share a neuron. So a neuron might sort of pull in signals from multiple receptors and send a summary to the brain. So it helps us save on travel space instead of paying attention to each receptor on its own. So low level processing at the level of the eyes. And then once we've had that low level processing, then we're going to send all that information through that optic nerve and into the brain. So our fovea, as we said, that's our focal point. That's our area of highest concentration. That's where we get the best detail. And then the optic disc is the technical term for the blind spot or basically the hole where our optic nerve is leaving the eye. So if I actually hop back to this image, you can kind of see that, so the retina is the back of the eye, and where this optic nerve is, that's where all of the neurons that are bringing signals from our sensors, our receptors, are going to be leaving the eye. And they have to leave through a space. They're interfering with where there could be other receptors. So this is technically a blind spot. And judging from where it is, not too far down from our fovea, it seems kind of weird that we would have a blind spot right in the middle of our vision. And if you've never done some of those little visual trick things, they'll usually print out a piece of paper and have a checkerboard on it. There'll be a little green dot and then an X, and they ask you to look at the green dot, but watch in your periphery as you move that paper until the X disappears. And you can then use something like that to see your blind spot, which is technically right in the middle of your vision. We don't notice it unless we're using one of those little trick images because our brain is really good at filling in those gaps. So we kind of ignore those gaps and we skim over them. So we're not getting information from there, but we don't really notice it. The same way that you don't notice that you can see the side of your nose all the time until, again, your professor draws your attention to it, and then you can be aware of that. Hmm?
Like you'll see squares inside like a bunch of stuff but like too much. Like is that your eyes? Like when people think your eyes playing tricks on you, so like obviously there's a reason. How did like those things work when you see like numerous dots?
Oh, this is one. We talk about a couple of illusions, but there are far too many that we can't talk about. Some of them play on psychology and how we organize stuff, our brain tries to find shortcuts. So we want to use as little processing power as we need to. So we skim over things. We ignore a blind spot. We fill in gaps in an image. We assume certain things based on experience. And so most optical illusions rely on the fact that our brain will try and fill in gaps. So that's like when you see the ones that are a whole bunch of squares or geometry shapes, and it shows like there are black dots on the page in between them, but there really aren't. That's because our brain is trying to sort of solve the puzzle that it's seeing, but our brains do it in a predictably and consistently wrong fashion. So we see things like those illusions. It's especially bad for perceived movement, especially on very busy images. We assume that things are moving, but that's actually sort of taking advantage of almost like those cicadas, those tiny little movements, and amplifying them so that we're finally aware of them. So there's lots of neat little tricks that our brains do unintentionally that optical illusion designers have figured out and can play with. It's a very cool topic. Yeah. Yeah. Yeah, it could be very similar. We're kind of, I don't know if I name it, but we talk about something pretty similar. There's a little bit of bleed over here with another topic, specifically on attention. And I have to double check. I think that comes into the consciousness chapter, but it really applies well with perception because it's all about what you are and are not aware of. But we talk about something called inattentional blindness, where if you're focused on something else, if you're looking for a particular thing, you will skip over stuff that doesn't fit what you're looking for. So I know it from foraging theory. So if you're a bird and you hunt black beetles, you have a visual representation of black beetles and you know about the size and shape they should be. Maybe you encounter a gray beetle, but it's not black, so it doesn't count and you ignore it. And it's kind of like that for us as drivers. If you're watching for cars, you only ever see cars. And so you can have issues where things like a bus or a motorcycle would then throw you off because it's not part of your search image. So yeah, very closely related to this stuff, but I don't think we ever actually name it in the course. So interesting to know. Yeah, as you can tell, these are one of those topics that I love, and I have to cut down how much I can talk about because we could spend an entire week on just this one yeah, so that's our blind spot. So a little bit of homework if you're up for it would be to look at so Google something like find your blind spot or blind spot image. They usually come up pretty easily. I used to have a textbook that had it, so I would get the students to like bring their textbook and do it, but I don't think our text has it. But it's interesting, and the first time you do it is always kind of okay, so the next thing to talk about, if we're talking about our retina, is what exactly are these receptors that I've been talking about? Because I've been intentionally vague, just talking about these sensory receptors and hand waving what they are. If you know anything about the anatomy of the eye, you might already know of the existence of rods and cones. Those are the two types of sensory receptors that we have in our eyes. So our rods are specialized for low light vision. They're really good at perceiving sort of black and white. They can tell the difference between dark and bright, and that's kind of it. They're very good for things like movement. They help us on our periphery. They give us really good detail for movement and things that we're not looking directly at, but they're not fantastic for daylight times. And in the daytime, we rely on our cones. This is our second type of receptor. And cones are for color. That's always how I remember it. The two C's go together. But cones do color and they're good at daylight vision. And the reason for this is in order to see color, we actually need a lot of energy to come into our eyes. So we need quite a bit of light, lots of brightness enter our eyes to get information about color. So rods don't need tons of energy because they're not looking for color. They're just looking for light or no light, hence black or white, light or dark. But the cones care about the exact wavelength that we're looking at. So we need a lot more light to be able to figure out the wavelength so we can turn that into an understanding of color. And our cones do a lot about detail. In fact, our cones are what sit at that focal point, our fovea, and give us the most detail when we're looking at our environment. So whatever you're looking directly at, you're using your cones for the most part to be looking at it. And we can talk about the process of adaptation, where we can become more or less sensitive to light as needed. My example that I use to explain this is movie blindness. So if you go to a movie theater at, say, noon, you walk in, you sit in a theater for 2 or 3 hours in the dark, you're relying very heavily on your rods to see because it's very low light conditions, it's dark inside the building, and then you walk outside and it's 2 or 3 o'clock in the afternoon and it's very, very bright. It's blindingly bright. You guys get all squinty, your pupils get really small to try and block out as much light as you can. And that's because our rods are used to having very low levels of energy, low light levels, and we have to switch from using our rods in the dark to our cones in the light. So we have this period of adaptation as we sort of accommodate and get used to it being a lot brighter. And the first, say, 90 seconds or so are about the worst of it. We get a pretty good changeover in the first 90 seconds, but to have a complete switch between rods and cones or back again, it takes about 15 minutes for a full swap over. So this is why when you go to bed at night and you turn your lights off, all of a sudden the whole apartment is black and you can't see anything at all, so you have to stand still for a few minutes. That first 90 seconds is when you have the fastest adjustment. After, 45 seconds or so, you can get by. And then by the time you're in bed, it's fine. But that's adaptation in action. So it works going from dark to light, but also from light to dark. So we have a swapping over process. All right, and I actually want to talk about things a little bit out of order. So instead of talking about this image here, I want to hop all the way to the next slide and talk about rods and cones, and specifically their concentrations in the eye. So this image here, this is a little bit of a weird one, but we're looking at, so we have our picture of the eye, and instead of looking at it from the side, this one is looking at it top down. So the lens is still at the front, but now our fovea is at the back, sort of behind. So we're looking down onto our eyes. And so here we can see where our blind spot is, where the optic nerve is leaving. That's at about 10 degrees, so 10 degrees over from our fovea. What I mean about it being sort of right in the middle of your vision, and we never know. And so as we go around the eye, all the way up to the sides, we can see 70 or 80 degrees to either side of our eye. And so that graph in the middle or on the side for us here, that's showing us what our concentrations of rods and cones are at all of those positions around the eye. So as we go up in degrees, it's basically wrapping around the back of the eye. So that's our retina and the wrapping around part. So it's a little bit of a weird graph to look at, but it shows us some really cool information. We see at our 0 degrees, that's where our fovea is, that's where that focal point is. We have a massive peak in the red. So that's where we have a very high concentration of cones. And that makes sense. Cones do detail, that's our focal point. We have tons and tons of cones at the place where we look at things, the place where we want the most detail, the focus. And as we get outside of that focal point, outside of the fovea, we see a dramatic drop off of cones, sort of down to almost none right away. And that kind of makes sense. But let's talk about rods before I talk about why it makes sense, because the rods pick up the slack. The rods would like to be in the middle, but they've been crowded out by all of those cones. We have too many cones taking up space in the fovea, So the rods get as close as they can, but the cones take the sort of charge of the situation. So that's why at that 0 mark, we don't have any blue. But just outside of the fovea, as we get just a little bit away from it, we see that really sharp increase in the number of rods that we have and a much more gradual decrease. So what that means is that we have more cones than rods at the fovea, but we have more rods than cones anywhere else. And I hope I didn't say it backwards the first time. More cones than rods at the fovea, more rods than cones everywhere else. And so when we look at this, we see that, yes, we have tons and tons of cones right at the fovea, but everywhere else, they're massively outnumbered by those rods. And our rods are actually a lot more present in the eye. We have way more rods than we do cones. So it kind of makes sense there that anything that isn't at our focal point, that isn't where we're looking intently, kind of relies on the lower energy requiring and less detailed rods. And this is why I was going to say it makes sense that as we get away from the fovea, as we get away from that focal point, we lose our color perception. So in your periphery, I can just sort of barely see my fingers here. My glasses don't help that matter. But on the edges of our vision, I'm aware that my hands are there. I can see them moving. But if I didn't already know what color my watch was, I might not be able to tell just from looking at the corner of my eye. I have to get to about here before I can see colors. So when it's just rods on the outside in our periphery, we don't get color detection. But that's where that movement detection comes in handy. And again, when we talk about attention, our periphery is very sensitive for something that isn't in direct focus. We are really good at identifying things that move or things that are shaped like biologically relevant items. So things like bears would definitely trigger us that we should pay attention to them, especially if they're moving, especially if they're moving towards us. So we'll talk about that attention drawing properties when we get into that topic a little later on in the course. Okay, and oh yeah, and this graph also shows us that the blind spot has no receptors, which makes sense because that is the gap through which everything is leaving. So you can't have receptors in a hole. Logical. Okay, so next we're going to hop back to this slide. And this is where we're looking at what actually makes up our retina. And I use the analogy of treating the retina as kind of like a projector screen, but that's not necessarily the best way to think of it because it's actually inside out to how you might envision it. So looking at this image here, we have light entering from the left and moving to the right. So that's the same as the eye that we were looking at before. The lens would be on my side of the screen, and then the back of the eye is on the other side of the screen. But you might notice that our rods and cones are the furthest thing away from where we would have the lens. So in order to get to those sensory receptors, light has to pass through all of the structures involved in supporting and processing information from those receptors. So in order to get there, our light has to pass through those ganglion cells and our bipolar cells and through the sort of connecty parts of the receptors, our rods and cones there, in order to get to the pieces that actually react and are photoreceptive. So this is one of those instances where if we were designing this intentionally, we might flip it around so that the light didn't have to travel through all of those structures in order to be picked up. But instead, we have it set up this way. So we pass past all of those things, we have to move around them to get to our receptors. And here you can start seeing why they're called rods and cones. The cones have sort of a tapered cone shape, and the rods are long and skinny. And here you can see that low level of processing that I hinted at. So we have those bipolar cells, and they're pulling information from 6 different rods. So they're going to be picking up on signals from all 6 of those cells and sending that combined signal to the brain. So that's that low level processing where they're going to add together or average out the firing from those receptors and send a combined, moderately processed signal out to the brain. In contrast, our cones seem to have a one-to-one setup. So that's what's going on there, and that kind of makes sense. The cones are more sensitive, they're giving us more detailed information. There are fewer of them, so they get that one-to-one processing for the most part. And again, this is one of those points where we could go into tons of detail about bipolar cells and ganglion cells and all of that stuff, but this is as far as I go into it. This is quite enough biology for one day. And so again, on that idea of let's gloss over a couple of the details, we'll talk very, very briefly about the visual pathway, but a lot of this is going to be familiar from what we had talked about when looking at the different parts of the brain. So we have here our left and right visual fields. So we have sort of the blue and the red. So we have the things that are on the left side and the right side. And we see that again, the red that is on the right ends up on the left side of the brain. And it's the red on the right of both the left and the right eye. So we get those right-sided visuals from both eyes, and they're going to end up on the left side of the brain. So the same way that we had crossover in our spine when we have sensory information coming in from our skin, we have that crossover, but here that crossover happens at the optic chiasm. It's going to cross over at that corpus callosum, specifically the optic chiasm part of it. And we're going to get all of the information on the right side of our bodies coming to the left side of our brain, and the left side of our sort of body is going to get processed on the right side of our brain. And we could talk about sort of the specific structures involved there, but we don't really need to know too much about it. What I will instead talk about are something called feature detectors, because these are way more cool than the sort of individual pieces of the brain involved. So feature detectors are basically individual neurons that will respond or fire aggressively in response to different features. So we have feature detectors that respond to lines, and specifically lines of differing orientation. So we might have neuron A that fires when it sees perfectly vertical lines. And if we change the orientation of the line, if we pivot it, we'll get to a point where neuron A no longer fires because it's pivoted so much that it's no longer vertical. But maybe now we have neuron B that fires for lines that are 5 degrees or more pivoted. Or maybe we have neuron C that fires for horizontal lines. We can also have neurons like this that fire for other things other than vision. There are really interesting ones that they've studied in rats that fire for particular body orientation. So if they face a particular wall in their enclosure, different specific neurons will fire in relation to their body orientation. That might relate to our sort of kinesthetic and vestibular info there as well. But for our visual stuff, we're focused in on sort of very specific features, things like lines or edges. We also have more complex ones. We have feature detectors that will fire when we see things that we classify as faces. So it's not just human faces. If we see an oval that has two circles at about eye level, that feature detector for faces will light up. It'll start firing to tell us that there's something that's kind of face shaped. And that's one of the reasons why people are so good at picking faces out of things that aren't really face shaped. They're just sort of vaguely the right proportions. Maybe you have a potato and it has lumps in the right spots. Your brain is sort of hardwired to look for faces because we have the hardware to tell us that it's kind of the right shape. So that's very interesting. And as we've had sort of massive advances in how we can record and look at neurons, we now have the ability to record a single neuron and all of its firing. So we can use that to look at these specific feature detector neurons and figure out how much do we have to rotate a line for that to no longer activate, and very cool things like that. All right, so as I said, a little bit biology heavy for the first part there. But what I want to do before we end the class is talk a little bit about color mixture. And the reason I'm going to do this is because we've talked about sort of our rods and cones and the fact that rods see black and white and cones see color, but we haven't talked about how we get from our whole spectrum of colors, how do we turn that into something that we perceive as the world around us? How do we know that the chairs are green and the ceiling is white? So how do we get to that point? And in order to learn about that, I want to talk about two types of color mixture. And that's because the type of color mixture that we're very familiar with, when you were in kindergarten and they did finger painting and they showed you that mixing two colors together would create a different color, that's subtractive color mixing, and it behaves very differently from additive color mixing, which is how light works. And because our eyes are dealing with light, it's going to behave very differently to our preconceived notions of color mixture. That's why we do this brief interlude to talk about color mixtures and additive versus subtractive. So that's why we're here right now. But as I said, what you're used to when you mix paints is subtractive color mixing. And the reason why it's subtractive is because of how we perceive light, how we get colors off of objects. So if you have red paint, that paint, it itself is not red, but it has absorbed all of the wavelengths of light except for red. So it looks red because it has reflected red wavelengths back at us. So it absorbed everything else and it sends red towards us. So it subtracted the blues and the greens and the yellows and everything that isn't red. Those all get absorbed or pulled out of white light, and we're left with red only. So if you mix red with blue paint being something that has pulled out all of the wavelengths except for blue light, is reflecting blue light back, we're then going to have sort of a mixture, and it's going to reflect a little bit of red and a little bit of blue. So we kind of get a bit of purple, which is what we expect. But we also need to keep in mind that it is pulling out everything. And the red paint might pull some of the blue that the blue paint is trying to reflect. And the blue paint might pull some of the red that the red was trying to reflect. And we end up with less light being reflected overall. Mhm. So that's like a technicality thing. So with light, so if we have white light is all of the possible wavelengths. There's all of the ones in between. So things look to be certain colors based on their reflective properties. So if you think of, I think it's like Vanta black, the one that absorbs all light, so it looks very, very black because there's no reflection. If we had something that completely reflected all light, it would look white because that's what it does. So they appear those colors because they reflect. So it's a technicality where it isn't red, it isn't the quality red, but it appears red because of other qualities. Yeah, I don't ask a lot of technical questions like that. You can go into a lot more sort of debate about that. But actually, we are very close on time there. But I would recommend if you have a chance, I have a video on additive versus subtractive color mixing. I find this does a great job showing the examples. And then I'll start here and refresh our color mixture series at the start of next class.